So this last talk of the morning is focused on a different approach that is also based in fluorescent correlation spectroscopy, but rather than focus on the diffusion, we will be focused on the occurrence of the concentration of molecules and the oligomerization state of molecule inside the cell. And this is also a technique that was developed by Michel and Enrico in the early 2005 or 2006, something like that. And it's called number and brightness. And well, if we want to understand, how is the what is the concentration in a cell, we can rely on different techniques. One example may be that, well, we have a cell and we can measure the intensity over time, right? In a point, I don't know, we make a box here or a box here, and we measure the number of count per second. And if we do a calibration with, I don't know, our label is green fluorescent protein, we can estimate that the concentration here, based on this calibration, can be, I don't know, 10 nanomolar or 30 nanomolar of EGFP tagged to a particular protein. And this is totally fine. It's something that many people do in the lab. But this does not allow you to tell if in this region of the cell, these molecules are, isolated one by one, moving around alone, or eventually they are interacting together and forming some oligomers, or doing some other kind of interaction, molecular interaction, that can change the brightness of the molecule. So for that kind of purposes, when we want to know interaction between molecules, other techniques like a FRET enable us to interaction between two molecules, and we learned yesterday that this is very specific tool to see if two molecules are together. But it has also some constraints, and you need to have, one molecule with one fluorescent protein and one molecule with another, and these need to occur in the space and need to be aligned in the space. So it has also some limitation. So there are, it's very sensitive to detect the pair formation, but has also some constraint. The other possibility to measure, determine the protein concentration and aggregation in the cell is, as Michel mentioned before, is the image correlation spectroscopy, in which if you have, situation where you have sparse distribution of a fluorescent molecule in your field of view, well, you can calculate the spatial autocorrelation function, and then you can obtain from there, for example, the autocorrelation. You feed the autocorrelation function, and then we can do all what Michel teach before my talk. But again, you have some constraint. If you have too many molecules in the field of view, you cannot do this spatial correlation, because it's based in the fact that you need to have isolated, molecule in order to correlate this in space. So and the other important thing is, if these molecules are moving too fast, you cannot do it too. So basically, it's important that you have, molecule with certain amount of movement. So the purpose of number and brightness is to provide, a pixel resolution map of molecular number and aggregation in cell. And the method came basically for the measurement of the first and second moment of the fluorescent intensity distribution. We will learn in a minute how it does. And it can be used, either in a confocal microscope, so a raster scan microscope, in a two-photon microscope, in a TIRF microscope, so based on camera, or a light sheet microscope based on camera, if you do the right calibration and the proper acquisition. The output of the method is basically to provide a map, so a sub-image, that give you the N, so the number of molecule concentration, and the brightness of the molecule, which is giving to you an idea if you have only molecules that are moving alone, so as a monomer, or some multimers of this. This can be compared in a plot, which is a histogram between number and brightness. And this is very useful to isolate, the different population of pixels you have in the cell. So basically, we will have some tools in order to use this approach, and depending if you have good detectors who are photon counting detector or analog detector, we will learn that we will need to do some calibration. But basically, the most important thing is, well, there is a software that can do everything for us and will give you the right values for the with the proper calibration for the number and the brightness of the molecule you have in our image. So this is the concept. So Michelle introduced us that if you have a point, right, and you measure this point over time, you will see fluctuation, right? And fluctuation can be, very, high or very small, depending on what? Well, depending if you have very large, amount of molecule passing through the volume or many, many more molecules, but alone in the same volume. So basically, the thing is, if you have in the volume the same amount of brightness in this and this, so you have the same amount of molecules in your excitation volume, but they are in some case forming aggregates, and in the other they are monomers. What you see in the trace of intensity is basically a change in the profile of the fluctuation. And if you think about if one molecule came in or out when you are in monomer, you have little fluctuation. However, if you have, oligomers that are going in and out, the amount of intensity that you are losing or gaining is quite high. So if you calculate the first moment, which is the average, and this is the formula, the K, and if you calculate the variance of the fluctuation, this is the second moment, you will see that the sigma here is way larger in the situation of oligomers in comparison with the situation of monomers, right? So if we can measure this variance, we can measure basically the state of the oligomerization we have within our pixels. And this is the trick. So instead of using the correlation to measure diffusion, we need to measure how much is the variation in the amplitude of the fluctuation, not in the time. So it's like putting our data collection in the other dimension, right? So when we do correlation, we care about how fast they move in the PSF, and if the correlation is long or is short. If the correlation is short, means that the molecule is very fast. If the correlation is long, means that the molecule is very slow passing through the volume. But now what we care is if the amplitude of this correlation is something or something else, right? And if we use this statistic, well, we can split, this information, and basically based on the ratio of the square of the average of intensity and the variance, we can obtain what is called the number of particles, so the concentration in the pixel. So these are the two measurements that we need to run. And in fact, if we do an acquisition for RIGS, it will work for NMB. It's not always true that we can do NMB and then apply RIGS, because for the NMB, it doesn't really matter to have a sober sampling of the PSF. We can have larger pixel, like, I don't know, 100 or 150 nanometer pixel for NMB, because we only matter about the fluctuation. But for the RIGS, it's very important because we need to, sample a very high, number of pixels in the PSF, because we don't want to lose of the molecule when they diffuse. In here, we don't care. So if you acquire your setup for RIGS, you can do NMB. The other way around is not always true if you don't have, if you don't want to do eventually RIGS, right? Okay. So the calculation, as I mentioned, are these, and we don't need to bother, but basically what we do is we calculate the variance, we calculate the average count, and then with these two variables, we can obtain the brightness and the N. And as you can see, basically the average count and the variance give it the number of and the brightness using this mathematical operation. So what we need to do in the acquisition is, in fact, acquire many frames, one after another, as quick as we can. And then the software will do the processing and will give you a plot with these two variables. An important thing of this is, well, in principle, we can change, this length of acquisition, so the dwell time or the frame time, and this will change the brightness, because if you stay in a pixel longer time, you will illuminate it more, you will collect more photons, and you will have, longer integration time. The problem is if you integrate for too much long time, you can end up having this problem in which eventually the volume where you are observing is homogenizing and you will never see the fluctuation. So when you have to run experiment with camera, for example, well, there are some, requirement in order to set up the experiment to proper collect the fluctuation that you need to measure, right? So these are the things that then we need to trick depending on the fluorophore that you're using, for instance, how much bright or not is, how much is how fast is diffuse or not, and etc. What are the contributors in the variance that you are recording with your detector? Well, there are basically two main contributors. And you need to know very well where are your contributors, because if you don't know, eventually you can end up, doing mistakes in the calculation. So the two main contributors of the fluctuation are your particles, the molecule that you are studying, and this is how the variance stand over the molecular brightness. So it's to the power 2. And then we have always noise from our detectors, no matter which detector we use. We have always some noise. But the good thing is that basically this scale linear, not to the power, so it's very easily to handle with one and another. And what we need to do is introduce both in our absolute variance. And if we do the proper calculation, then we can obtain the true number and brightness for our molecules and obtain in our image the true oligomerization that we have and the true concentration we have pixel by pixel in the image. Okay. So this is how we calculate the N and E. And basically, if you operate this mathematics, you end up having the N here and the epsilon here. And basically, these are the variables you measure. Basically, this is the trace of fluctuation in a pixel over time. And this is the variance of this fluctuation over time. So these are the variables that we measure in our image. And then we operated them in order to acquire, to calculate these two parameters. And that's it. The software does the rest of the work. Okay. So the thing is, there are dependence, as I mentioned, of the brightness to the molecular brightness to the variance, and this scales to the power of 2. So it's a quadratic dependent, and this is an example. It's a calibration done with EGFP in solution, and you see that if you increase the power, you will see that you will go up and up and up. And this is the function that describes this behavior. And it's something that you know very well. And it's something that you have to calibrate into your experiment. And this tells you something more. So if you do an experiment and eventually you have solution where you start with a cell and you set up your power and the whole experiment was running okay, but you move to another cell and now you are saturating your system and you attempt to reduce the laser power, or your calibration is already screwed. So you need to calibrate again, because the amount of power that you will introduce in your sample, it will give you the absolute brightness you get in your molecule. It doesn't mean that you will get basically different state of oligomerization, and we will see in a minute. But basically, you need to be very careful in the calibration. And in every set of experiment, you have to calibrate that. And in every day, you have to calibrate so you can compare one day to another. And this is how important is also the mobile to the immobile fraction. And Michelle was explaining what it means, mobile and immobile fraction. Immobile fraction is a dye that cannot diffuse. For us, if the dye cannot diffuse, it will not fluctuate. So basically, no matter how many counts you add by increasing the power or whatever, and this is logarithmic, so you are going for 1,000 to millions, you see that the power increased, but in black, the slide, which is, some of these chroma slide, stay flat. And the other important thing is if you do the proper calibration for each of the measurement and you do the EGFP, you will get, a value for the brightness, which is flat over a range, and at a certain point, it's going to drop because you will start to saturate your system and it's no longer linear, right? And this is basically the range that you need to know. I can work in my system between this range and this range, and this proves that basically you can measure your system with any amount of laser, but the brightness will be always the same, right? Okay, so this is an example. This is an example in which we have a solution of fluorescent beads and fluorescein. Fluorescent beads are quiet during the acquisition, while the fluorescein can diffuse all over because it's very fast molecule diffusion. And if we calculate the brightness and we calculate the counts, you can see that we have generated this plot of intensity over brightness. And if we select the value of 1, which is the value for something that not fluctuate, right, is the immobile fraction, you will select the region which are basically the bits. While if you select the ones that give you higher brightness, so you select basically the fluorescein that you have here. And if you have a very good, system like a photon counting detector, and you have, molecules that can form only oligomers, only monomers, only dimers, only tetramer, or only octamers, you will have, a plot in which you can see here, this is the monomer, the dimer, the tetramer and octamer, and they are appearing and step which are proportional to the previous one. Let's say if you have, a brightness of 1.2 for the monomer, the expectation for the brightness of 2, right? So a dimer will be 1.4 and go on and go forth, right? So 1.8 and 2 point something, right? So this is the thing. And you will go here and select a region of interest here, and you will trace back what are those images, those pixels on your image. So the other important thing here is that the brightness and the number of molecules can be measured independently. And this is, again, a calibration that is done. You can see that here, in the histogram of intensity over brightness, you have a cloud a little bit above of 1. And then you can see the linearity between count per pixel over the brightness. You can see also the number of particles over concentration. So you have a long range, which is linear. And then you can see that the brightness versus concentration is also flat, which means that no matter how much dye you have, if the dye is monomer, it will not give you all the results. You can have more and more and more and more counts, but the brightness will be still flat. So it will still giving you the right information. And this is a calibration that was done to prove, this situation. Okay. But most of us do not have, or we don't rely on truly photon counting detectors in our instruments. Why? Well, because they are expensive. They have some other issues in order to work, etc. And at some point in the lab, Michelle, Enrico, Claire, and many others discovered that basically, if you do the proper calibration with any analog detector, you can do the same calculation. And these are the parameters that you need to measure. So the parameter that you need to measure in order to understand if your analog system will work doing number and brightness or not is this. You need to define what are the digital levels, or what are the increments, that are, absolute numbers that give you, this brightness or this other, right? You need to define what is the offset of your detector, and you need to understand what are the variants at very low current when, I don't know, you turn off the laser. You have electronics now, so things like that. Okay, if you do that, and you can calculate this offset, the digital levels parameter, and the variance of your analog detector, well, you will include all this information in your software, the SimFCS, and then it will give you the right number and the right brightness of your sample in any way. And this is how it look like. So what we do is basically we acquire the noise of our detector. And after that, there is a function that obtain the data, what is the amplifier Gaussian noise, what is the potential distribution of digital level, and what is the offset. And by doing this fitting in your data, you will introduce these values in the formula, and the formula will do the calculation, and then we will have this data ready for our measurement in our experiment. Well, this is another calibration using EGFP, and see how linear is the brightness on the laser power. You see that it's very, very straight. And how flat is the number in regards of the laser power, which is all important, because otherwise you will get different results depending on how power you have in your sorry, in your experiment. And this demonstrated that we can use basically with the proper calibration almost any microscope, any commercial microscope you have in your lab. And this is a result indeed, in which they did, the fluctuation of each EFP in CHO cells. And you see that the distribution of the fluorescent protein is different, right? The amount of fluorescent that you have here or here is quite different. If you do the map of number, you see, well, yes, here there are more concentration than here. I don't know, you have 50 molecules here and you have, I don't know, few molecules here, right? But if you do the molecular brightness, the cell is flat, because each EFP is usually monomer in cells. You don't have each EFP oligomers in cells if you don't tag each EFP to something else. So each EFP like to be as a monomer inside the cell. And you can see that here you can, obtain quantitative volume on about the distribution of concentration in your cell, at the same time that you can get how is the oligomerization state in your sample. So to close basically this presentation, I will show you an example, which is basically a follow-up on the RIGS part of the paxillin, which is a very important, protein related to the focal adhesion, as Michelle showed us. And in this project, which was a big consortium in the LFD and at the 2000s, probably, right? To understand the cell migration, there was many groups. It was one of these big grants. They developed these tools, the RIGS and the NME, to try to understand and to tackle how was the migration of the cell, and try to understand how was the dynamics of some molecules who were important in the movement of the cell over space and time. Okay, so basically the question was, how is the molecular dynamics of the focal adhesion, and if they tag paxillin with each EFP, do we have within this focal adhesion monomers, oligomers, or what kind of blend between monomer oligomers we have? This was the thing. Okay, well, they did this acquisition, so they acquired, I don't know, 100 frames at certain size of pixels, pixel size. And then after that, they measure the average intensity, and you can see that focal adhesions has high concentration of paxillin, but paxillin has also, high concentration in other regions of the cell, besides of the focal adhesion. And in other regions is low concentration. But if we calculate the B map, so the brightness map, we realize that, for instance, I don't know, all these regions here where we have a huge change in intensity is flat in the brightness, which means that the protein is monomer all over there. However, in the focal adhesions, you see that they are in increasing number of molecular brightness, which is telling us that there are some sort of oligomers. And if you look carefully, there are some spots here who are white. And Michel introduced in the previous talk the concept of immobile fraction. So these spots are regions where there is no fluctuation. When we have no fluctuations, we cannot see any brightness, any numbers, right? But it's telling us that basically this focal addition has a degree of oligomerization, and perhaps inside there are oligomers that tie so high that they cannot, move easily within the time that we are acquiring our data, okay? So if we go to our plot, the variance versus intensity, we can set what is the monomer, and after do the calibration, right? We calibrate our experiment measuring EGFP free in the cell, and we obtain this level of brightness, let's say 1.2, or no, 1.2, no, it's 1.0 something in that case, right? And then we look at where those pixels are in our image, and you see that this is more in the cytoplasmics, right? But also we can go to the higher oligomeric state. If we have, if we are lucky with our system, and if we have the proper calibration, we can go to the specific oligomer, the dimer, or the tetramer, or the octamer. But if we don't care and we want to just understand if there are oligomers or not, we can just select a region which is above, the monomer range, and you see immediately that this pop up information in the focal additions, and as we saw before, you see, region where there are holes, and these are the immobile fractions. We can also use in the software, this kind of a Gaussian distribution to select, monomers or oligomers. Basically, in this cluster, the calculation was that oligomers are basically most of the time octamers of the bacilli. Okay, and this is basically the set of data that we use for analysis. So from frame 0 to 100, 100 to 200, 200 to 300, etc., go on and go forth. And you see that from beginning to the end, there are regions where it was very high in intensity, and it was going down over time, like this one. And there were some others that don't even exist at the beginning, and it was increasing over time. And there was another which were flat. The interesting thing here is, over the years, we have learned that basically these focal addition are really high dynamically. And if you introduce in the system the right stimulus, you can form and ensemble this focal addition over time. And using a set of set of data, we can chop this time set of data and analyze this in terms of number and brightness over the time very easily. And this is a proof of how this is unfolding. So you see, this is the intensity over time, and you see that the intensity starts from, I don't know, this point and came to the half. In this other region, remain quite flat, and in this other region, start to grow up. So this was a proof that the dynamics of the focal addition was very, very high. So to conclude this presentation about this new method that we can use, I insist, on top of the rigs, if we acquire data for rigs, is interesting because we can put another level of understanding of our system. We can understand basically the number and the brightness of molecule in each pixel. And using this, we can basically compute the concentration and the oligomerization we have within the pixel. And it doesn't mean that within a pixel you can have not only just one kind of oligomer situation. You will have mixture, and this is the reason for why it's very hard in cells to obtain only dimers, only tetramers. You have mixture, but what we are measuring is how is this variance, and this variance is different from when you have Y more monomers than when you have Y more oligomers, right? And the other thing that is important here is that we can remove or take out easily the immobile fraction, which will not contribute in our experiment. And this is important because it can bias our result. And I hope I can demonstrate with the example of the Paxilin that number and brightness is very powerful, and it can give you results not only in space but also in time. So you can have, I don't know, 1,000 frames, and then you can analyze little part of these 1,000 frames, and then you can put information on top of this. So with that, I think this is the end of the talk, and now we have time for some questions.